Welcome back to the Poor Proles Almanac. Again, my name is Andy, your host, and today we're joined by Dr. Liz Koziol, an assistant research professor at the Kansas Biological Survey and Center for Ecological Research at the University of Kansas. Dr. Koziol's work is at the forefront of understanding plant-microbe interactions with a particular focus on arbuscular mycorrhizal fungi and their application in grassland restoration. Her research includes comprehensive assessments of commercial microbial inoculants, which has garnered attention from NPR in recent memory. In a recent study examining 23 products, Dr. Koziol's work has highlighted issues with product label inaccuracies and low viability in commercial products. Beyond commercial products, Dr. Koziol is a pioneer in using native mycorrhizal fungi to improve ecological outcomes. Her studies show that introducing native fungi can significantly enhance native grassland diversity and suppress weeds, with the benefits persisting for several years, as we'll discuss. This approach is crucial for advancing native plant restoration, improving the establishment of desirable species, and accelerating plant succession. Her research has also demonstrated the practical benefits of native fungi in agriculture, showing positive effects on crops like tomato and pepper varieties in organically farmed soils. So with all that said, we had to talk, and her research is illuminating. So take a listen, let us know what you think. Liz, thanks so much for joining us. You've really carved out a unique space for yourself around prairie restoration and native microbial inoculants. So please tell us a little bit about yourself, how you got interested in this very specific space in ecology.
Sure. Well, I'll introduce myself first. I'm Liz Koziol, and I'm assistant research professor at the University of Kansas, and also the curator, one of the curators of INVAM, which is the largest collection of mycorrhizal fungi, the type that I study in the entire world. And I also have a small business called MycoBloom. And so I've always been drawn to the natural world, especially the things that are ephemeral, the things that emerge and disappear with seasons, like morels, for example, in the woods, or asparagus that grows huge in your garden and then disappears below ground until the next season. It's just beautiful. And I guess what really led me to ecology was, I'm inspired by the natural world, but it wasn't a clear plan. For me, it was kind of more the people that I work with. So after college, I was working with the developmentally disabled, and I took a somewhat random hourly job in a lab that happened to study the type of fungi I've ended up devoting my life to, my career, which are called arbuscular mycorrhizal fungi, which is a mouthful. And I usually just call them AMF or AM fungi for short. And so in this hourly job, I really fell in love with research, discovery and connection to the land. But more than that, I formed a really close bond with the lab leaders, who are Bieber and Peggy Schultz. And today I'm still working with them almost 18 years later. We're still collaborating, and it's grown into something more like family, our relationship. And so what is really unique about the research in Jim and Peggy's lab, they built this living collection of AM fungi from old growth prairies. And they use this collection to explore a wide range of ecological questions. So they started studying how these fungi influence plant establishment and succession in the prairie. And my dissertation work built on their foundational research, and I started asking questions that are more applied about what particular AM species might benefit plants the most, how do we deliver these fungi effectively, how do we optimize their use in restoration. And my perspective was using these fungi in a restoration context, at least initially, although throughout my career, I've started looking more at perennial agriculture and annual agriculture and some other sort of crop species too.
Awesome.
That's how I got started. Yeah.
Yeah, that's quite a ride. So what exactly makes the arbuscular or AM fungi special?
Yeah, so they're symbiotic with plants. That's really the heart of why they're so special. They form relationships with about 80% of terrestrial plants, it's estimated, including almost everything we eat, most crops, most herbaceous species like grasses and wildflowers, and some trees. And so unlike a lot of people, when they hear the word fungi, they think of a big, beautiful mushroom. These microbes are subterranean, so they spend their entire life cycle below ground. They do form that hyphal network below ground, but rather than making mushrooms to reproduce, they have little teeny tiny spores below the soil surface. And what they're really known for is these fungi are crucial for phosphorus uptake, but they do more than that, including influencing the water relationships of the soil and that plants have with soil. They can turn on plant immune systems and help suppress pathogens, and they can influence plant competition. They can even affect the nutritional quality of plants, for instance, the protein and calcium content in the crops we eat, and the phytochemicals that plants produce, such as CBD concentration in hemp and latex in milkweeds.
Oh,
They don't just support individual plants, they can shape the entire communities and influence these long-term ecosystem trajectories. And so these aim fungi, there's around 350 species described right now, but there's likely thousands of them. They're really pretty hard to characterize because they're microbes and they're subterranean. And so for a long time, we didn't even know they existed.
yeah, it's interesting. You've kind of focused, I know you said you're starting to transition away from or towards perennials and agriculture, but you've done some work with native prairie restoration, which I think everyone would agree probably needs to be done, but it doesn't seem like it really gets the traction that reforestation gets or something like that, or defending forests. There just doesn't seem to be a draw towards the prairies in the same way, for better or worse, mostly worse. So how does this kind of fit into that restoration work? Like you'd mentioned, like that trajectory for an ecosystem.
Yeah, I'd say I agree with you there that prairies don't get the attention they deserve. Some people, they have this disease we like to refer to as plant blindness. So some people see a yard and they call that a grassland, versus some people see a highly diverse prairie ecosystem of 350 species and they call that a grassland, you know? So it kind of depends on people's perspective, and a lot of that is education about what makes these ecosystems so unique and so awesome. And a lot of that to me is focusing on the diversity of the prairie ecosystem. I will say, although prairies haven't got the attention they deserve, they're getting more attention because of things like pollinators. People are really excited about pollinators. You know, there's this huge movement across the US and other nations about the monarch butterfly. And so that's got people to think about milkweeds and wanting this plant, which used to be this, you know, it just can be toxic to cattle, this weed that they wanted to get rid of out of their yard or out of their land. People are starting to support that plant because of pollinators. And so in some ways, prairies are getting a little bit more attention because of these unique plant species that they have and the benefits they can provide to ecosystems, you know, both within and outside of the prairies. And so that's exciting for prairies, I think. There are some movements to protect these spaces. There's different groups. Kansas has some, there's some in Missouri. Throughout the range of the prairie, there's different small groups who focus on acquiring old growth prairies. That's another term that is often more commonly used in forests, but for sure they're old growth prairies. Some of these systems have been in place and maintained by Native peoples for, you know, tens of thousands of years to be managed as a prairie. And so protecting these spaces is a goal of some of these up and coming groups, and seeing that is really exciting.
Yeah, it raises a really interesting question. I think for somebody with like a rudimentary understanding of ecology, it's typically framed like early succession, quote unquote, is bacterially dominated, and then late succession is fungally dominated. And then typically on that matrix, early succession is always seen as kind of like grasslands or prairie. But that, you know, to your point, there is old growth prairie. So where does fungi kind of fit into that narrative of, well, technically this is for old growth forests?
Mm-hmm. Great question. Yeah, so prairies wouldn't be prairies without fire. And so we've used fire to manage these systems for a really long time. Native peoples have been doing it, you know, consistently, basically from Canada all the way down to almost Mexico in the central US was a prairie. Now it's mostly corn and soybeans, but you can imagine all those spaces that are corn and soy were prairies for the most part. And fire is essential to these systems because it gets rid of the woody vegetation that might take over the early successional forest species like mulberries and, you know, cottonwood. So if you don't put fire in the system, it's quickly can change into this more shrubby, short trees or early successional tree environment. And so fire is a really useful tool. Some places you can't burn as easily as others. So when I moved to Kansas, they burn everything all the time, and it's awesome. And I really like fire, so this has been exciting for me career-wise to get to burn my prairies. So more in Indiana, where I did my dissertation research, didn't burn as much. Some places they do, but they would use mowing as an alternative to management to get rid of these woody species. So maintaining the prairie does take work. In terms of the relationships with mycorrhizal fungi, some of my research, my first or second dissertation experiment, looked at how different plants across a successional gradient in a prairie, so all prairie herbaceous grasses or forbs, how they responded to these old growth fungi. And so we collected these fungi from an old growth prairie and put it with different plants across the successional gradient. I had 25 in my first study and looked at the plant response. We call this the mycorrhizal response. We had pots with the fungi and without. And essentially, these early successional plants, things like Rudbeckia hirta, which is the black-eyed Susan, they really don't respond to native AMF that much. They don't care about them that much. And in contrast, our later successional species, our Morphicanescens, our lead plant, our Echinacea pallida, pale purple coneflower, they tend to be really, really sensitive to these native AMF, and they tend to really like them and do a lot better when they have their old growth fungi with them. So across the successional trajectory, fungi become more and more important for these late successional plants, which are often our restoration targets.
It's interesting. I think that probably means that there's a lot of incomplete research right now on the relationships between fungi and species because of the way we understand its relationship to kind of that, to your point, that woodier community, because it's supposedly they're more co-evolved to break down woodier materials and so on. But clearly we don't know everything at this point. There's something possibly very significant that we haven't figured out yet.
Yeah, clearly. I think currently we have a database of 202 plant species where we've looked at this mycorrhizal response. But of course, in the prairie ecosystem, there's, hundreds more than that. So we haven't even looked at all the possible plant species for sure. One relationship we have sort of picked out that is related to this mycorrhizal response is plant root architecture. So many of these later successional species tend to have coarser roots, more perennial roots. If you think about the Asclepias tuberosa, the butterfly milkweed, tuberosa, tuber, you think about like a potato, it has this really thick taproot and very few fine roots. And so this plant and a lot of late successional plants like it, without these fine roots, they're really dependent on mycorrhizae to use their hyphal network to do the stuff fine roots would do. So there's this trade-off. They invest in these fungi to use that hyphal network to collect phosphorus and water and those things from the soil. And they produce fewer fine roots. And that's in contrast to our early successional species that are less dependent on fungi, and they have a ton of fine roots, generally speaking.
Yeah, that's interesting. Now, with AM fungi, you always refer to as native. So is it actually native in the sense that this type of fungi is only found in the US, or is it because it's only been harvested there? And is there any data to compare that to, like, if it exists in Europe or wherever, to say it is the native one that's particularly important?
Mm-hmm. Yeah, good question. So what is native in an AM fungus? So for almost all of my research experiments, I've worked with what I call native AM fungi to do restoration experiments and sometimes agricultural plantings. And so for me, what that means is I'm collecting fungi from an old growth prairie that's within around 50 miles, usually it's closer, to the site I'm trying to restore. And so I'm using a really locally adapted fungi that probably would have been present in the space I'm trying to restore, but, of course, has been lost through land use changes and anthropogenic change. That's what I'm doing for my research. In terms of how local a fungi has to be considered native, that's a open-ended research question. There's been some studies on that. And we do know, like, fungi from Illinois work differently in Kansas and vice versa. We've done some of these trials, but we don't have a great answer for that.
Sure.
And in terms of how widespread these fungi are, that is also a great question and something we'll have to do a ton more research to figure that out.
Awesome. Yeah. Not to get into the nitty gritty too much, but like, what does your harvesting process look like for that AM fungi?
Yeah. So our process to select these fungi, we go to an old growth remnant, So it's a native habitat that's never been plowed. So that's a major condition that we have. So it's never been plowed. It's been prairie for as long as it's been prairie, tens of 10,000 years potentially. What we do is take a soil core, which is hollow cylinder. It's about 1 inch wide and it goes usually about 18 inches or so. And we can pound that into the ground and collect, the top 10 inches of soil or something. From there, we can actually find a ton of spores of these mycorrhizal fungi. We can get a fungal community from this old growth prairie with just essentially like a cup of soil. It's really hard to actually isolate the fungi out of that soil. So that process is essentially running that soil core through sieves, which are like a colander you would use to get your spaghetti out of the water. But the sieves, instead of a big colander for your spaghetti, are on the microscopic scale, like 0.001 millimeter scale. And so we target the size of the soil using those colanders that are the size of these fungal spores, which I can send you a picture of their spores. They're really beautiful. And so the spores are sort of like their seed. It's their dormant structure and has a lot of genetic material. And the color and shape and size can be really useful in determining the species of AM fungi. And so from that soil core, out of that sieve, we can target spores. And then in the microscope, we spend tons of hours sorting them by different color and shape and size. And there's some other traits we look at in terms of the morphology of the spores to separate them out into species. The challenge with growing these fungi is that they can compete with each other. So if we want to grow, let's say 10 species from a prairie, we have to grow them all separately. Another challenge with these fungi, which makes them hard to work with, is they are symbiotic. So they cannot actually grow and reproduce without a plant. And so we have to grow the fungi with the plant in a sterile media for about 6 to 8 months. And then when we actually want to use the inocula, if we want to use a diverse native mixture from Prairie A, for example, and we have 10 species from Prairie A, we would, at the end of the growing season, combine all those cultures up together to make a diverse mixture that we would apply in the field.
Yeah, that's awesome. And you figured out that this is actually something we can utilize and market, which is really cool. So can you talk a little bit about like what drove you to say, you know what, I should sell this and people really should be using it?
Well, great question. I'd say my family has some entrepreneurs in it. Growing up, my dad used to take me to the flea market every weekend, and then he was an electrician with his own business. So, seeing people create businesses, kind of out of nothing, was something that was ingrained in me since being a kid. So it wasn't too scary for me to think about starting a business. But what really led me to create it was the market gap. So we've done all this research, both mine and other people in Jim and Peggy's lab, about the benefits of native fungi, how they can promote succession and inhibit weeds, and especially these old growth prairie fungi. And so we presented all of our talks, for years, for decades even, and land managers and nursery professionals were starting to learn about these microbes and that they help, but they didn't really have access to them. Commonly I'd be asked like, this is great, but like, what are we supposed to do? And the answer was pretty unsatisfying,
Yeah.
Like basically, if you know a prairie that's going to be destructed, try to save that soil and use it, was a very unsatisfying answer. And so I started MycoBloom because I became really good at propagating these native mycorrhizal fungi, these old growth fungi, via my research. And I figured it's not that much more of a step to try to do this outside of the teeny tiny pot scale to bulk propagate these for people, restoration practitioners who want to put these microbes into work in the field, at scale. And so it was really the research inspired the people, and the people wanted a tool. And then I said, well, I think I can give you that tool. So there was MycoBloom. Yeah. Mm-hmm.
Yeah, that's awesome. And you've also done some assessments with other commercial microbial inoculants. It's really interesting. I don't want to take your thunder away a little bit on this. So could you talk a little bit about what you found and what the real world consequences are for people that are buying these?
Yeah, so I've done quite a few studies with commercial products up to 10 years ago in native ecosystems. And most recently, I've done some work looking at U.S. commercial inoculants. There's a study in applied soil ecology where I looked at 16 commercial products And another study in New Phytologist where I did a meta-analysis, which means taking data from anything that's been published and reanalyzing it to see if there's common trends. And so that data set had different scientific studies looking at commercial inoculants from across the globe. I think there were 4 different continents represented in that study. And both studies, both the US and this global scale study, found pretty similar findings, which were commercial products are really lacking quality control. Most of them aren't viable, or the spores aren't viable at the time that these end users were using them. The scientists were trying to put them on plant growths and see if they infected plant roots and if they benefited crop growth. At the time of use, most of these products were not viable. Even more scary is that multiple studies have found that some of these products are including pathogens or maybe other filler organisms that they don't provide benefits and worse, can even harm plants. This is a huge problem, and it persists because there's really no global standards, and in most places, no mandatory testing before products can hit the market. So yeah, that's a major challenge.
Yeah, and it's not like those are cheap products either.
Yeah, they can cost up to hundreds of dollars for, a few ounces of this stuff. And so another role that I have, in addition to being a scientist and being at MycoBloom, is that I'm a curator at INVAM. So INVAM is the world's largest collection of arbuscular mycorrhizal fungi, which are the type that I use in my research. And we have around 1,000 isolates from across the globe. And it's a nonprofit housed at the University of Kansas, and it's awesome. And so through INVAM, we provide AM cultures to researchers or industry, really anyone, and technical advice and quality control assessments for industry as well. And that's been really enlightening to me, working with industry in that role. One thing that really surprises me is how many inoculant companies out there are operating without any internal staff who actually know how to assess the quality or viability of mycorrhizal fungi, which I think is pretty insane.
Yeah, I don't mean to pick on anyone in particular, but the trajectory of cannabis grower to influencer to seller of microbial products is not insignificant. And not to say that you can't do anything in your backyard and learn, but to your point, when you're talking about something at this small of a scale, it's a little bit beyond the scope of a backyard gardener. And to your point, if you're shipping products across the globe, you really need to have that kind of stuff dialed in.
Yes, absolutely. Currently in the US, there's almost no regulations on these products whatsoever, including enforcement of what people put on labels. In some places, like California, has a specific regulation on what has to be put on a label and how you have to describe your species and the number of spores that are in there versus propagules, but that's California specifically. And the reporting mechanism for companies is you can self-report your own product concentration and quality. And so there's there's really no oversight in that. And you can see why that could be a problem if there's not a third party actually also looking at these products. Because some companies, there's really no incentive and no one there to check them to say, look, you said you had, 40,000 spores per gram, and there's zero. There's really very few people to do that, So it's kind of fallen on the scientists' shoulders, which is why I did the study to look at some of these products and to see that on average, reported spore concentration or propagule concentration was only about 2% versus what was claimed on these products. So that's insane to me.
Yeah, absolutely.
And some products claiming and having zero. Yeah, it's a huge and massive problem, and we need some regulations out there for these companies.
Yeah, and it raises some really interesting questions about the guidance for using these products when they have so little quality control, right?
Exactly.
And that's something you've researched extensively as well, is how do we actually utilize these to best benefit the ecosystem that we're applying them to? And you've shown some really interesting stuff about how using these fungi can improve diversity, suppress weeds. I'm curious about what that looks like and maybe if it has any role in dealing with like invasive species pressure.
Yeah, so from a restoration context, I've definitely been trying to fine tune how we apply these inoculants. So I've done a number of studies where we put the different inoculants on the roots of seedlings, or we call them plugs, before we transplant them
Hmm.
out into restoration. And that method works fantastically. And that's because we can use a very small amount of inocula, the smallest amount of inocula, put it on the roots of a late successional prairie seedling. We know that seedling will be a really great host for our AM fungi. They grow together before we put them into the field, and we're already sure that, that symbiotic relationship is in place. And so we found that method of moving microbes into restoration to be extremely effective in many restorations across the tallgrass prairie ecosystem. But of course, it's really hard to transplant a bunch of seedlings. So some places use seedlings, other places do a more broadcast or drilling seed based approach if they're doing restoration at scale. Like, for instance, I did a 15-acre restoration this spring with Johnson County Parks. And, planting plugs on the scale of 15 acres would be a lot of work,
Yeah.
massive amount of work. So, of course, in that
Mm.
situation, land managers at that scale are more broadcasting or drilling seed into the upper surface of the soil. And so, thinking about applications for those types of projects, one thing I did in 2016 when I moved to Kansas, I set up an inoculation density experiment where I applied different volumes of a native old growth fungal inoculant into a field, ranging from none, which kind of represents our traditional seed-based restoration projects with no microbial amendments, to sort of middle amounts, what we see on the recommendations of most commercial products, and then really high inoculation rates, which actually kind of reflects what science use. They tend to use inoculation rates that are much greater than what's reported to be used on commercial products. So I wanted to capture that whole range of like how much of this stuff do we have to put out there to see an effect. And so I've been
Mm.
following this study. Now it's in its ninth growing season, which is awesome. And another problem we have with these AMF application studies is that oftentimes they don't follow them for very long. So this study is really cool and unique in that we have a data set that ranges up to, this is going to be its ninth growing season. And so what we see is that the more of this native AMF applied in the field, the more of these late successional plants are established. And probably because of that, there's this indirect effect where these late successional plants take up the space, they fill up the niches, and then there's fewer non-native plants and weeds that are allowed to establish because those native plants with their native fungi are really competitive and they can beat even the strongest of the weeds for the most part.
That's awesome.
So yeah, we've seen this sort of pattern time and time again in different application strategies. But yeah, really fine-tuning not only how much do we need, but also what tools do we need to put it out there. So I have a grant submitted now where we're looking at different application strategies. So some places don't have a drill for seed, and so they can't drill an inocula. So maybe those places want to broadcast, and then what piece of equipment and at what rate do we have to apply it? So there's a ton of research, and it takes a really long time, especially in prairies, to see some of these effects.
Yeah, but you're doing the work and that needs to be done.
Trying. Mm-hmm.
I feel like you can go down these rabbit holes. Like I live on the edge of the Pine Barrens, and you just see this rare ecosystem getting eaten up by invasives. And the problem is, like, you cut down an autumn olive, and the microbial life around it has fundamentally shifted in a way that limits the potential for native species to reclaim that space. And it's so difficult to try to wrap your head around that. So it's nice to know that you're kind of getting it on the opposite end too, right?
Mm-hmm.
Where you've got these really healthy microbial, old growth AM fungi that are in there, and they're actually resistant to that invasive species pressure.
Mm-hmm.
That it kind of swings the other way too. It's really hopeful, I think, for folks that are like looking at all these different pressures hitting these old growth prairies, that they have some tools to resist against that pressure.
Mm-hmm. That hits to a point that I just want to toss something in there that wasn't on your question list, but that's thinking about like the dispersal of these organisms. You say that you brought up that example of the autumn olive and how the soil is changed in that area, which can inhibit native plants. In restorations, we're often working in these spaces that are post-agricultural land spaces. So they've been tilled, for in some cases hundreds of years. The mycorrhizal communities are decimated. And so in this long-term study that I put out there, it's year 9. The plots are 3 meters from each other, so 10 feet, a couple big steps. And we're still seeing this effect of that initial inoculation event almost 10 years later, which to me means that these fungi are moving really, really, really slowly. So they're not necessarily going even 3 meters away after a decade. And these fungi are pretty dispersal limited. So one of the ways they move
Mm,
around is crawling in their hyphal networks below ground. That can be really slow. They can move around, in the guts of worms. That could also be really slow and sporadic. And they can move around on our little, dirty critters down there, our star-nosed moles or whatever that move little soil particles around. But yeah, their
mm.
dispersal is really, really limited, which is one of the reasons why these applications of native microbes are really effective.
Have you looked into, and I'm not sure if you're familiar with these at all, like Korean natural farming or like jadam as a dispersal technique to share these or, get these out there further or to maybe democratize access to something like these microbes?
Yeah, I don't know too much. I've heard Korean natural farming. I've heard that phrase before. It's kind of like fermenting sugars. Is that?
Utilizing sugars to harvest microbes in a specific, usually old growth ecosystem.
Yeah, I don't know too much about any research in that realm, to be honest, but
Yeah,
yeah.
it's very DIY trying to get local microbes as opposed to your point, these very often ineffective microbial solutions you can buy online. It was
Hmm.
a way to try to improve the ecology and usually post-agricultural or agricultural space through these co-evolved microbes that have been in this region for X amount of years. And obviously, there's no real knowledge of what you're harvesting, but at least it is theoretically, if it's coming from an old growth area, something pretty good. Fingers crossed.
In theory, it should be there. Yeah, if it was there in
Yeah.
the past. Yeah.
And I'm thinking about like for folks that want, that are interested in restoration work, even on a much more granular scale, maybe, they can afford to buy, seeds for some of these old growth prairie plants and want to try to create a small, maybe a 10 by 10 ecosystem in their backyard. What kind of tools are available to kind of like get this type of stuff in the hands of regular people?
Yeah. I think about the native seed industry a lot because I think they're a great example of, groups that have really, really optimized how to use and acquire local biota. And so,
Mm,
30 years ago, there were really no native seed companies in the US. And today, in the tallgrass prairie, in some states, there's dozens of these little companies and nurseries that are selling local ecotype native plants harvested from remnant prairies nearby. And then sometimes they grow the seeds directly, and sometimes they might cultivate them for a year or a few years, in a farm. And so I think that's really inspiring. And I don't see any reason why inoculant industries can't mirror that approach of like, of course, native plants and locally adapted plants, people understand that and they think that's important. And I'm telling you, it's also important for microbes. And so, but getting people to see and experience the microbes is much more challenging than looking at happy and not happy plants, because they're from the range or not from the range are supposed to be. Um, Observing that in fungi is much more challenging. So there's a little bit more legwork to do. And
mm.
potentially, there might not be 30 different companies in a given state selling locally adapted micro products, but I think it's crazy and extremely surprising that, I've been doing MycoBloom for 10 years, that really there's no other companies out there who are even thinking about providing locally sourced microbes. I'm just disappointed with the other business owners out there and people who work with microbes that they're not trying to start this, in their country or in their region. There's been talk of people trying to do this, but it is actually pretty challenging to grow mycorrhizal products because they are so sensitive and require microscopy skills and laboratories and, molecular tools that most people don't have. So to do it right, to have a product label that's effective would take all of those resources. But for, home growers, one thing that I could say can work if you don't feel comfortable using, products of microbes out there or MycoBloom's not in your range and you don't want to move foreign microbes to your space. What you can do, maybe not dig up an old growth ecosystem. I would never recommend that.
That's what I heard.
I would never do that unless it's slated for destruction. Then, yeah, of course, dig all that up and use all that great soil. But what you can do, let's say you're planting a restoration and you've tried this before and you have a Baptisia, a plant that is doing really well. You can harvest some of that soil around that plant in your own yard and use that as inocula for the next round, for example. So, yeah, if your plants are doing well, then they don't need any mycorrhizae, right? Or other
Mm,
microbes. So think about that. If you have plants that are doing well, you can use their microbial communities to help your new plants. That's one thing that, the average home grower can do. And I've worked with some people in Kansas who they got the message with MycoBloom, they got the message with native microbes being really important, and they've done exactly that. They went to their successful restorations and used some of that soil as inocula for a future restoration. So I think that's really inspiring that it's catching on in the private sector too.
yeah. Now, looking ahead, where do you see research going? Where do you think the research needs to go around utilizing AM fungi and maybe, its role in both restoration and agriculture?
Mhm. That's a great question. So there's research for research's sake, which I do some of that. And there's lots of people who have small fungal collections that they use in their labs for research. And that's one thing, but it's often really different from what the commercial industry is offering. And I think it is important to continue doing research on these commercial products as well, both in terms of identifying which ones are useful or not useful, but also looking at how how these products are sourced, where they come from, this effect of being local or not local, and what this might mean in a restoration context. Because generally, if people are going to use microbes, they aren't going to be harvesting old growth soil and they aren't going to be digging up their successful restorations. Let's be honest, they're probably going to be buying a product. So I think it's really important to look at these products in ways that we can make them better. I wish industry would listen to more of the research, which shows things like diverse microbial inoculants are much better than these single strain products, which is mostly what's being sold. Many of the packages have one or two different species of fungi. But when we're doing our native restorations, we're using, sometimes as many as 30 different isolates of AM fungi. We know that works really well. Diversity generally is really good. We want diversity in our prairies. We should also want diversity in our AMF communities and including our AMF products that we're applying out there. So thinking about diversity and local adaptation, I think is really important. And just finding how many species of AMF we have out there, looking at some of these old growth systems and characterizing the fungal communities there hasn't really been done for many of our ecosystems in the US, including lots of different teeny pockets of remnant prairies that exist across this whole gradient, from Canada to essentially Mexico. And so some researchers have done it in certain places, but it's not been a vast sweep of what communities are there. So in terms of what to restore, we don't even know what's there. Characterizing what's there and protecting what's there, I think is really important.
Absolutely. So, for folks that want to buy your products or read your research, where can I send them to?
The easiest list is my Google Scholar page. If you Google Scholar, my name is Koziol. I have a website I can link to. MycoBloom has a web page as well. A lot of my research, almost all of my papers except for one I'm a middle author on, is open access. I think that's really important.
Mm,
My work is really applied, both in prairies and in agriculture. And so I want people to, be able to read how I applied these ideas and my findings. And so I try really hard to make all of my papers open access so anyone can read them. So they're all out there if you Google me.
I enjoyed the fact that did not have to use Sci-Hub once to find your
Yes,
papers. So I appreciate that.
it's really important to me. Yeah.
That's awesome. Liz, this has been fantastic. Anything you wanted to mention that we didn't talk about?
Well, I just want to share a story,
Yeah, yeah,
a
yeah.
quick story. So last weekend, I was at a native plant sale here in Lawrence, Kansas for the Grassland Heritage Foundation, and I had a vendor booth for MycoBloom. And a few people came up to me saying, like, hey, did you hear that all these commercial products are total crap? And I'm standing there like, yeah, because I'm the scientist from the NPR story that's been playing in my community for the past 2 months who just told you that. That was my research. And so, because of my research on commercial inoculants, a lot of people have lost faith in mycorrhizal products. And I get that. And that's totally valid. I don't have much faith in them either. But being commercial isn't like it's not automatically bad. There's a lot of bad actors out there. But, in VAM, we've seen these really weak products, but we've also seen some good ones. And at MycoBloom, we're trying to do something different, something that's grounded in ecology. And we're designing our inoculants with restoration in mind, using native strains, using high viability, high genetic diversity, and fungi that are really beneficial to their native plants. And I think it's a great example of how the industry could work, but we do need standards for sure, both for MycoBloom and all the companies out there. Yeah, it's hard to both say that commercial products are terrible and also have a commercial product.
Yeah, that's fair.
It's challenging, and I get why people are scared of commercial products. And yeah, I'm one of the people telling you should be scared of commercial products. But fungi are amazing. They do amazing things in restoration, and they do also great things for a lot of the crops we grow. And so I think don't totally lose faith in mycorrhizal fungi as a tool. I think we can get there collectively, but we all need to pay a little bit more attention to make it an effective tool.
And we'll get there, I think. Just, some bumps along the way.
Yeah, absolutely.
Liz, thank you so much. This has been fantastic.
Thank you.